Dear student, today we will study kingdom Protista. Protista, they are eukaryotes, which include all eukaryotic organisms that are not animal, plant and fungi, mostly unicellular, but some are multicellular. They are mixotroph. Locomotion over here is by three different locomotory organs: flagella, cilia and pseudopodia. Flagella is a whip-like appendage attached to the body. It turns and twists in the water in order to push the body through the water. Now if you look at the structure of flagella in protist, it is different from the flagella which is present in the prokaryote. The small basic difference is that the basal structure of flagella in case of protist is present in cytoplasm, whereas in case of prokaryotes it is present in the cell membrane. Then there are protists which bear more than one flagella. Cilia, they are short, fine hair-like structure present all over the body. Pseudopodia, they are protoplasmic extension which help in movement and capture of the prey. Now mode of reproduction, both asexual and sexual reproduction is seen. Asexual reproduction is by three different methods: binary fission, which could be longitudinal or transverse, budding and multiple fission. Sexual reproduction is by formation of gametes, and the fusion of gamete is called syngamy. Here syngamy is by three different ways: isogamy, anisogamy and oogamy. We'll just briefly go through all these mode of reproduction. So in case of binary fission, from one cell two different cells are generated, either longitudinally or transversely divide. So before the division, its DNA is duplicated and one more copy of DNA is generated. Then other cell organelles also divide or duplicate, you can say, and then cell membrane pinch off and two daughter nuclei are generated. In case of budding, there is a small extension growth in the cell, and later on the DNA duplicate, one of the copy goes to that extension, and then it is enclosed by a membrane, and then again at the maturation two different cells are produced from one cell. In case of multiple fission, there is one cell and the nucleus undergo several divisions. So many nuclei are present, and these are now walled by cyst formation. The nuclei later on develop their wall, or you can say membrane and cytoplasm, and then when the favorable condition comes, the cyst break and daughter cells are released. Usually this mode of reproduction occur under the unfavorable condition. Syngamy is the fusion of gamete. Now fusion of gamete which are morphologically same but physiologically different is called isogamy. When the fusion of dissimilar gametes, dissimilar in many aspects, takes place, then it is called isogamy. Oogamy is kind of anisogamy. Oogamy is kind of anisogamy in which fusion of male and female gamete occur, where male is motile, small and active, and female is non-motile, large and passive. Next is lifestyle of protist. The sexual reproduction involve alternating haploid and diploid stages. Now in haploid cell, one set of genetic material is present, whereas in case of diploid, two set of genetic material is present, one from either parents. This is called haplontic or haplodiplontic life cycle. Now just have a brief idea of haplontic life cycle. It is with zygotic meiosis. The organism over here is haploid, multicellular, and it produces the gamete. This is haploid, it produces the gamete. Gametes are always haploid. Here no meiosis is required. So without meiosis it is generating the gametes. Now the fusion of gamete occur and it produces zygote which is diploid. Now this zygote will undergo meiosis to produce the daughter cells which are haploid. So this meiosis is called zygotic meiosis. If you look at here, diplontic life cycle, it is with gametic meiosis. The organism over here is diploid. It undergo meiosis and produce the haploid gamete. So this is called gametic meiosis. And later on they fuse to produce the zygote. Now this zygote will not undergo meiosis, but it will undergo mitosis and produces the daughter cells, which are diploid. So this is the life cycle. Again, in different protists, different kind of life cycle is present. Another feature of protist is presence of contractile vacuole. Now what is contractile vacuole? This is a type of vacuole which is present in protist and help in osmoregulation. Osmoregulation means maintaining the electrolytic balance to maintain the osmotic pressure in the cell. As the protist habitate in water, so they need mechanism to maintain constant osmotic pressure in the cell. In freshwater protist, the cell is hypertonic as compared to the surroundings. So water enters cell by endoosmosis. If the water continue to enter the cell lysis may occur, and to prevent this, to prevent the cell lysis, contractile vacuole is present. The contractile vacuole stores and remove the excess of water by contraction. Also it helps in the excretion, that lot of waste is removed along with the water. Contractile vacuole is absent in marine protist as they are present in hypertonic surroundings. Now let's see the classification of protist. Now this classification is actually based on the mode of nutrition. So photosynthetic protists, which are known as phytoplanktons, they are autotrophs. Slime molds, which are saprophytes. Zooplanktons, which are also called as protozoans, they follow the heterotrophic mode of nutrition. If you see their further groups, phytoplanktons are divided into three: dinoflagellates, chrysophytes and euglenoids, whereas saprophytes are acellular and cellular type. Zooplanktons, amoeboid, flagellated, sporozoans, ciliated. Now we will discuss them one by one. First, let's go through the dinoflagellates. Now over here I'll be only discussing the unique features of dinoflagellates, not all the characteristic. So I'm not discussing very general characteristic, only specific characteristic. They possess a cell wall like structure as a outer covering, which is called theca or luria. This is composed of cellulose. The theca is divided into several plates by giving an armored appearance. It means what? The cell wall is not continuous, but it is divided into several plates. Usually they have two flagella, longitudinal and transverse. The transverse flagella transversely furrow between the wall plate, just like this. You can see transverse and longitudinal flagella. They appear in different color in sea and give different appearance to sea, such as red tides, night sparkling, etc. This is because they exhibit phenomena of bioluminescence and phosphorescence. Some of the dinoflagellates are colorful due to the presence of pigment and are associated with coral in sea. So they are symbiotically associated with corals and they give different colors to the corals. Let's understand what is the difference between bioluminescence and phosphorescence. Bioluminescence is the light emitted by living things through chemical reaction in their body. So they produce the light by the chemical reaction, whereas biophosphorence is the absorbance of certain wavelength of visible spectrum. Means the light which is absorbed is actually emitted. So this light is actually absorbed by fluorescent protein such as luciferin, okay, in cell, and emission of light occurs at lower energy level. So let's see some example over here. Gonilex species, they all exhibit red tide. and that is sudden increase in their number in sea surface, and it makes the sea appears red. Gonilex Polyda and Gonilex spinifera. Gonilex catinella produces toxin which can kill the fish and paralyze the prey. So some of these species are also toxic. Noctilica scintillans, the night light, they exhibit the phenomena of bioluminescence. To catalyze the reaction, they contain a specific protein called luciferin. What is their ecological importance? That they do the carbon dioxide fixation in the ocean, so that lot of other organisms, they can derive their nutrition from them. Now next is chrysophytes. Here we complete with dinoflagellates. Next is chrysophytes. They contain mainly diatoms and dasmids. Characteristic, again unique features I will discuss. They are freshwater as well as marine. The chief producer of marine ecosystem. They consist of cell wall which is thin, so they overlap each other. The cell wall consists of silica. So here cellulose is not present. Thus the wall are indestructible because silica is the resistant, you can say, material. It forms the chemical which is resistant to so many things. When the diatom die, they leave behind silica cell wall. These accumulate over millions of years and lead to large deposit of silica on ocean floor called diatomaceous earth. It may extend for several hundred meters, which can be mined. Let's see the significance. They are believed to produce about 50% of the world organic matter just by doing the carbon dioxide fixation. Diatomite, which is obtained from the diatomaceous earth, it is a porous and chemically inert, therefore used in the filtration of sugar, alcohol and antibiotic. It is also used for making insulating bricks, insulation of refrigerator and furnaces. It is used in toothpaste and paint to increase night visibility and as a filler in battery boxes. The fossilization of diatoms also form petroleum. Euglenoid, they are freshwater, marine and also found in damp soil. Instead of cell wall, they possess protein rich layer called pellicle, which is flexible and allow the change in the shape. Cellulose is absent over here. They are mainly photosynthetic but also follow heterotrophic mode of nutrition. They have two flagella, one small and another one long. The short flagellum terminates over the base of long flagellum, so not seen protruding out of the body. Now if you look at the structure of Euglena viridis, then there is no cell wall. Pellicle is the outermost, which is made up of protein. It makes the euglena flexible. Then all the cell organelles which are present in the eukaryotic cell are present over here, and extra contractile vacuole is present. Then over here you can see the flagella, and where this flagella ends there is another small flagella, and at the bifurcation photoreceptor pigment is present and it is sensitive to light. It guides the flagella. Then here is the eye spot. It consists of lipid droplet and which function as a lens. So this is the structure of euglena. So here we complete with the photosynthetic protistians. In the next video we will see the rest of the classification of Protista. Thank you.